Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about overview of the both prokaryotic cell and eukaryotic cell. So now in this lecture, we are starting with the prokaryotic cell and we will study about all the features corresponding to each and every layer that we have studied, and not only for these layers, and inside the cytoplasm also some components are present for each and everything. Slowly we will study about all these things. Now in this lecture, so mainly we will study about what are the various examples for the prokaryotes and what are some important features that were mentioned in NCERT regarding the prokaryotes, that also we will study. And then what are the different shapes of bacteria. So 4 different types they have given. So those also we will study, and then we will study about the cell envelope first. So in that cell envelope also mostly we will study about this glycocalyx layer will be completed, and also we will study about some information related to cell wall. Okay, so according to the lecture time and duration, so we will study up to that particular part. Clear? And now we are quickly going with this, and the first thing we need to study is examples. What are these examples? Now let us see here. So prokaryotes, what is the major information regarding the prokaryotes? First let us quickly go through this. We are studying about prokaryotic cells. Prokaryotic cells. First we will see about what is the meaning of this. Already in the previous lecture also we studied about the meaning. So prokaryon. So karyon is the nucleus. And here in the case of these cells, true nucleus is not present. It is the pro means what? Before form. Because in the evolution from the prokaryotes, eukaryotes were evolved. In the eukaryotes, true nucleus was there, but here it is a pro, before form. Here complete nucleus is not present, nuclear membrane is not present. Anyhow, these features once again separately we will talk maybe in the next lecture or after that. So when we are specifically talking about cytoplasm and its components, at that time we will study about the DNA, nucleoid and all the things. At that time once again we will study about this particular point. Is it clear? So that's why it is the pro, before form for the nucleus, and these cells do not have well defined nucleus. That's why these particular cells are called as prokaryotic cells, or simply these organisms are called as prokaryotes. That's it. Okay. And what are the various examples that we are studying under prokaryotes? The first thing that was given in NCERT is bacteria. First I will give NCERT words and then we will go for further information. So they have given bacteria, and then they have given blue-green algae, and then they are talking about mycoplasma, and PPLO they have given. PPLO. So already we have studied about these names, right? What is this PPLO? It is a pleuropneumonia like organisms. Pleuropneumonia like organisms. Why they have given the name like this? Pleuropneumonia like, because there are some organisms that are causing pneumonia, pleural pneumonia. Pneumonia is the infection to the lungs, right? So they are causing some infection to the lungs. That is called as pneumonia. So these organisms, see, already I told you this, PPLO are nothing but mycoplasma, right? So within the mycoplasma, these are a specific group, and these PPLOs, they are actually appearing like the microbes that are causing pneumonia. So that's why they are called as pleuropneumonia like organisms. This is what the name that they have given for this particular PPLO. Fine. And these are the 4 examples that they have mentioned in NCERT. Now we will go with clarity here. Bacteria. Bacteria means what are these bacteria that are coming here? Already we talked about this. Eubacteria and Archaebacteria. Eubacteria and Archaebacteria. So both of them are part of prokaryotes. Next one, blue-green algae. Blue-green algae are also called as cyanobacteria. These are also called as cyanobacteria. Understood or not? And next point is mycoplasma and PPLO. Both of them are same only. Yes or no? Mycoplasma is a broad name, and within that PPLO are a type. So that is the reason why when we studied about the size of the cell, at that time already we studied that mycoplasma are the smallest cells having the size in the range of 0.3 micrometers. And within the mycoplasma, a specific type is there that is called as a PPLO, and they are having the average size of 0.1 micrometer. Yes or no? So that's why we are saying that mycoplasma are the smallest cells. Mycoplasma are the smallest cells. Even though viruses are smaller than these, are you considering viruses as living organisms? No, we are not considering them as living organisms or cells. So that is the reason why we are excluding the viruses and we are considering mycoplasma are the smallest cells. Is it clear? This is what some basic information that we need to understand. And here, so all these things, all the prokaryotes will belong to only one kingdom, that is kingdom Monera. All the prokaryotes belong to only one kingdom, that is kingdom Monera. Fine. So for this Monera, so we are talking about all these things. Anyhow, I already told you in the previous lectures also. There is a separate chapter called as a biological classification, and there you will study some other informations related to kingdom Monera and all these things. And there you will study some other information. And here in this chapter, our main focus is to study only the structure of the cell. Yes or no? Structure and function, that's it. Only these parts we are focusing here. That is the reason why we are not going into internal details of all these other components. So what is this particular prokaryote and what is its function and how it is related, what type of infection it will cause. These things we are not focusing in this particular chapter. In this particular chapter, our aim is to study only structure and function. That's it. Only these things we will study in this particular chapter. And that too, I already told you and I made it very clear that whenever we are talking about prokaryotes, and in the general sense we are talking with respect to eubacteria, you should not forget this. This is very, very important. Whenever we are talking about general features of prokaryotes, always we are talking with respect to eubacteria. Understood or not? If they want to deliberately ask about this, then they will clearly mention whether it is eubacteria, archaebacteria or whatever it is. Understood or not? So that is the reason why. So from now onwards, whatever the general information we are studying, so mainly with respect to eubacteria only we are focusing. Okay? Unless and until we are specifically talking about so and so. Until then, don't talk about those specific organisms, so that it will be with respect to eubacteria only. This is a very, very important thing, because generally since now you are studying about this particular chapter, so you will remember. But after 1 month, after 2 months, after 3 months, after 1 year, again you will forget about this specific point, and unnecessarily you will think about, so they will give one general character with respect to prokaryote, and you will think that in cyanobacteria it is not like this, in archaebacteria it is not like this, in mycoplasma it is not like this. So don't think like that. So general features we are talking with respect to eubacteria only. Understood? So that is the reason why we are studying like this. So this is the first point. So in the first point they are talking about some examples. What are the various examples for prokaryotes? 4 examples they have mentioned in NCERT. Bacteria, blue-green algae, it is also called cyanobacteria, and then they have mentioned mycoplasma and PPLO. Though they have separately mentioned this, both of them combinedly they will come under a single group, that is called as mycoplasma. Clear? That is the first sentence. And then they are talking about the size. So next one they are talking about the size. Already we studied about the bacteria size. So in the theory part they have given 3 to 5 micrometers, and in the diagram they have given 1 to 2 micrometer. So that's why we have combinedly written this as 1 to 5 micrometers. This is the size. Understood or not? So this is the size, average size for a bacterium. This is what we are talking. But here you need to understand this sentence with respect to prokaryote. They are talking the sentence in NCERT with respect to eukaryote. With respect to eukaryote means, now we just see here. Suppose this is a prokaryote, that is a bacterium. Let us see about the eukaryote, and here let us take one human cell. Suppose any cell you can take, no problem. What is the average size we studied for a prokaryote or bacterium? It is in the range of 1 to 5 micrometers. And what is the average size we studied for eukaryotic cell? It is between 10 to 20 micrometers. Yes or no? Now, based on these values, at least, already we studied these values. Based on these values, now you just tell me which one is smaller, which one is larger. So now definitely you can say that prokaryotes are generally smaller than eukaryotic cells. This was the sentence given in NCERT. So prokaryotic cells are generally smaller than eukaryotic cells. Understood or not? So this is what the sentence that they have given, and these are the values already we studied. In this way you need to interlink the content, so that you can easily understand the NCERT statement. Understood? So this is one particular one. And now they are talking about another information. What is that information? They are saying that they will multiply more rapidly. and this, what is this, more rapidly with respect to multiplication. So multiply more rapidly than eukaryotes. What is the meaning of this sentence? Prokaryotes are multiplying more rapidly. More rapidly means fastly. So prokaryotes, they will divide very fast when compared with eukaryotes. This is what the sentence, very simple meaning, you can easily understand this, but we need clarification from the NCERT. Yes or no? And what are the various examples that were given in other chapter? Please remember, in this chapter they have not given any example, but in some other chapters they have given the examples. So what you need to do is, in the NCERT, anyhow, while reading NCERT also I will tell you, so wherever that sentence was given, just beside that, in that free space, you need to write the information. What is that information? Please understand here. So now we are talking about name of organism and what is its division time. Division time. Why you are talking about division time? Multiplication means what? One cell has to divide and it should form two cells. And what is the time taken for this process? What is the time taken for this process? That time is called as division time. And this division time will tell us what, whether it is fast or slow. Understood or not? If the division time is less, multiplication is fast. If division time is more, multiplication is slow. Yes or no? This is what the conclusion. So we need to write some examples of organisms that were given in the NCERT but in some other chapters. So based on this time, we will decide so whether the statement given in NCERT is true or false. Understood or not? So this is what I am saying. We don't need to study anything out of NCERT. Simply we will study from NCERT, but what are the different sentences we need to interlink so that we can understand the concept very clearly. Okay. Now here we are talking about total 3 examples. First I will write the examples, names of organisms. First one we are talking is Escherichia coli. Escherichia coli. Okay. So this is a bacterium, and this name is a scientific name. Whenever we are writing the scientific name, we need to write in italics. Since we cannot write in italics, we need to underline this particular words separately. This is the genus name, this is the species. Okay, so each word should be underlined separately. Anyhow, regarding this in the biological classification, so in that chapter, so you will study about, so what is the scientific way to write all these things. So everything we will teach you. So now let us go through this. So Escherichia coli. So underline each one separately. Fine. Now this is one organism. Within brackets I am writing. This is the scientific name, okay. But what is this particular organism? It is a bacterium. It is a bacterium. Bacterium means it is a prokaryote. So one example for prokaryote we are writing here. Now we are going with another example. What is this particular example? It is Saccharomyces. This is also scientific name. Saccharomyces. Saccharomyces. It is the genus name. So we are underlining separately because it should be in italics. And then species name. It is Cerevisiae. Cereviceae. Saccharomyces cerevisiae. This is the scientific name. And what is this particular organism? It is a yeast. It is yeast. Okay, this is the example that we are talking. And the next one is human skin cell. For the human skin cell we are talking here. Okay, in general we are talking about human cell, but in this case whatever the time we are writing, that is applicable for skin cell. Okay, because even in single individual also different cells will have different times. Anyhow, regarding this particular point in the cell cycle cell division chapter we will discuss more. So simply here we are writing human skin cell. Fine. Okay, so now let us go with the timings. What are the timings for this? That means from one cell, 2 cells should be developed. So for this particular division, what is the time it requires? That is called as division time, and you can also call it as generation time. It is also called as generation time. To form the next generation cells, not individual, for the formation of next generation cells. In this case, they are single celled, so that's why you can say next generation organisms also. But for human organism means it will be different. So that's why for the cell to make the next generation cells, what is the time it takes? It is called as a division time, or it is also called as generation time. Anyhow, these names in cell cycle cell division chapter also we will study, and now we will directly write the answer here. What is the time it takes for E. coli cells, E. coli cells. In short, if you are writing E. coli, Escherichia coli, bacterium for the division, it is taking 20 minutes. The time it takes for division is 20 minutes. And for Saccharomyces cerevisiae, it is yeast and it is taking approximately 90 minutes. That means in 90 minutes, one cell will become two cells. And here for E. coli, one cell will become two cells in 20 minutes. This is the meaning. And for human skin cell, for the division, that means from one cell to form two cells, what is the time it takes? It is taking 24 hours. It is taking 24 hours. If you are writing full, you need to write HOURS hours. If you are writing in short, then you need to write simply H. You need to understand these also. Okay. In the physics, you will study about units and dimensions. So there you will study about what is the scientific way of representation. Please understand. Suppose you are writing about the gram. You want to write about a gram. Okay. Suppose it is 2 grams of so and so you need to write. So you can write full like gram gram, and some students are writing like gr, some are like writing like gm, and some are writing like g. And which one is correct? What is the scientific way of representation? It is the correct one. If you want to write like this, you need to write like this. So in this way, you need to understand these also. You may think that so they will not matter. No, they will also matter here. So everything is important. And some students, they are having the habit of writing every time capital letter like this. And for some are having a habit of writing everything small. So don't write like this. First you need to understand, because some things should be represented like this only. Some things should be represented like this only. So what is the standard way? Learn it and accordingly you should use it. Okay, start using this. It is not just that you are studying it for examination purpose. You need to use it also. Okay. So anyhow, this is what the information that we need to understand. Now the next point you need to understand is, out of these three examples, which one is prokaryote, which one is eukaryote? That is also important, because that is where we are start comparing. Now Escherichia coli is a bacterium, so it is a prokaryote. It is a prokaryote. Okay. And then what about the Saccharomyces cerevisiae yeast? Yeast means it will come under the fungus. Fungus means it is eukaryote. So it is eukaryote. Eukaryote. And next one, human. Human means kingdom Animalia. Kingdom Animalia means once again it is eukaryote. It is eukaryote. Okay. This is the information. And now, in which chapter they have mentioned this particular one? Though now at this point you are not worried about the chapter name, but I am telling here how we are interlinking the information. Regarding the Escherichia coli, and it takes 20 minutes. This information was given in class 12th, and there is a chapter called as a molecular basis of inheritance. And in that there is an experiment that is called as Michelson and Stahl's experiment. In that experiment they have given this information. So it is needed here. So that information we need to use here. Okay. And next one. Regarding the yeast, it is taking to 90 minutes, and human skin cell or human cell taking 24 hours. These two points were given under cell cycle cell division chapter, and that informations we need to use here. And now you just see here, this is the example for prokaryote, and these two are examples for eukaryotes. Now you just compare the values. Which one is taking more time? Eukaryotic cells are taking more time, whereas prokaryotic cell is taking less time. What is the meaning of this? So if the division time is less, what is the meaning of it? They will form the next generation cells very quickly. That means prokaryotes multiply more rapidly than eukaryotic cells. This is the sentence given in NCERT. Now you understood? So this is what the explanation we want. So you need to read NCERT sentence, you need to understand, and what are the interlinking points. Once you've done all these things, preparation is completed. So we don't want to go out of NCERT. Whatever we are studying, everything is from the NCERT. That's it. So in this way, what is the complete sentence they are saying? Prokaryotes are generally smaller than eukaryotes. That is the one sentence. With respect to size, they are talking. Okay. With respect to size, they are saying that prokaryotes are smaller than eukaryotes. And prokaryotes multiply more rapidly than eukaryotic cells. This is what the sentence. Understood or not? So this is the second sentence that they have given. And after this sentence, now they are talking about the shapes of bacteria. What are the different shapes of bacteria? This is also very, very important information. What are the different shapes of bacteria? Different bacteria are having different shapes. And here we are talking about four major types. Four major types. I am saying that it is not that only four types are there. Not only these four types, many other types are also there, but the major ones that we are generally observing are these four types. Understood or not? So based on this, now this is the NCERT information, and the same information now we are converting into a table. So what is that one? First we will talk about what is the shape of bacteria. What is the shape of bacteria? And based on this shape, what is it called as? Based on the shape, what is it called as? So in this way, we are making a small table. And now we will talk about the first one. Some bacteria are rod-like bacteria. That means they are appearing as rod-like. Rod-like means they are present like this. Rod-like bacteria. Okay. So some bacteria are appearing as rod-like, and if they are present as rod-like bacteria, and this rod-like bacteria are called as bacillus. Okay, plural bacilli, singular bacillus. Okay, so they are called as bacilli. Understood or not? Next one. Next one. Some bacteria are spherical. Spherical. Spherical means like this, round in shape. Understood or not? So in this way they are present. If the bacterium is having the spherical shape, and this type of bacteria are called as cocci. COCCI. Cocci is the plural. If it is singular, it is coccus. Within brackets, I will write COCCUS, coccus. And here the singular is bacillus, BACILLUS, bacillus. In this way, we are writing this. Understood or not? Whereas some bacteria are having comma shaped, some are having comma shaped, comma shaped. Okay. So what is this comma shape? Generally, this is the comma. So some bacteria, they are having the shape like this. And if they are having the comma shaped, and then they are called as a vibrio. They are called as vibrio. Understood or not? This is one important point. And the next one is regarding spiral shaped. Spiral shaped. Spiral shaped means what? Like this they are present. Understood or not? This is called the spiral shape. And if they are present like this, they are called as a spirillum. Spirillum. These are some important points that we need to notice. Understood or not? So in this way, in the NCERT, they are talking about mainly four different shapes of bacteria. Based on the shape of bacterium, so different names were given. So now this information, directly they may ask that, so rod-like bacterium is called as, anyhow four types we are studying, four options you can get it. So directly they may ask this. So what will be the answer? Bacillus. And the spherical shaped bacteria are called as coccus, whereas comma shaped bacteria are called as vibrio, and spiral shaped bacteria are called as spirillum. Direct questions. And sometimes they will give a matching. So they will give a pair, and you need to identify correct or incorrect pair. They will give bacillus hyphen rod-like, and then coccus hyphen comma shaped they will give. Now we just see, is coccus comma shaped? No. So it is incorrect pair. So in this way, identify correct pair, identify incorrect pair. Like this also they may give. Or else they will give these on one side, column 1, they will give this on other side, column 2. And now you need to match this. So in this way, different questions are possible on this particular information. So it is a very, very important information. You need to compulsorily understand these particular names and meanings. Clear? And here generally, whenever we are studying about this, generally we will talk about some different types of examples. But in my opinion, examples are not needed. Unnecessarily don't waste your time, energy, memory on these particular unnecessary examples. Okay. So with this particular part is completed. But now I am giving one simple example. So based on that example, now you need to understand this particular one. Okay. Now here in the class 12th, there is a chapter called as a molecular basis of inheritance. In that chapter, we will study about an experiment. That experiment is called as Griffith. Griffith is name of scientist. So Griffith's experiment we will study. Based on the name of scientist, we will study about Griffith experiment. Okay. Anyhow, 12th class is not completed for you. So since you are 11th class students, you don't know about this. So that's why I'm directly giving the information. What is the information? In this experiment, this scientist used one type of bacterium. What is that bacterium? That bacterium is Streptococcus. Streptococcus pneumoniae. Pneumonia, N-I-A-E, pneumonia. Pneumonia is disease, pneumonia is the species name. Since it is a scientific name, we need to underline this separately. Okay. So now in this particular one, and this is the microbe or the bacterium that was used by the scientist. And now tell me, now the question is possible here. What is, anyhow I have given the basic information, so now I am asking the question. What is the shape of bacterium that was or what is the shape of bacterium on which Griffith performed experiment? Like this the question can be asked, or else the same question can be asked in a different way. What is the shape of Griffith's experimental organism? In his experiment he used this one, so you can call this as Griffith's experimental organism. So what is the shape of Griffith's experimental organism? Is it a bacillus or a coccus or a vibrio or a spirillum? What is this? So now understand this. And here if you are studying, anyhow this name was given in NCERT in the class 12th, not in class 11th. So anyhow when we are studying about that particular chapter, that day also once again I will tell, so don't worry. So here I am simply telling about the information, how to understand the NCERT, that's what. What is the meaning of strepto? Strepto means it is a chain. It is a chain, okay, continuous chain like. Chain like what? Coccus. Just now we studied coccus. What is this coccus? Is it rod like or spherical? It is a spherical bacterium. It is the spherical. It is a chain of spherical bacteria. And what is the disease caused? Pneumonia. So based on this they have given the name. So anyhow this name is not needed now, and we will focus on this. What is the chain of spherical? This is the chain of, it is a single spherical bacterium, and here the chain means they are present like this. In this way they are present. Understood or not? So in this way they are present. It is a chain of spherical bacterium. That's why it is called as streptococcus. Understood or not? In this way, based on this word also, there are different names are there. Okay. So diplococcus. Suppose anyhow, this information whatever I am teaching now, so it was not mentioned in cell chapter. For your understanding purpose only, I am telling you this. Okay. So here it is out of NCERT. So simply some names are there here. Diplo. If you can understand the word, you can easily understand this. It is not a difficult part. Diplococcus. What is the meaning of diplo? Diplo means two are present. What are these spherical shaped bacteria? Yes or no? So how they are present? Like this they are present. It is called as diplococcus. And in the same way, streptococcus. Just now we studied. What is this? Strepto means it is a chain, continuous chain of coccus bacterium. Streptococcus. So how this will appear? It will be appearing like this, continuous chain. Understood or not? In the same way, here it is staphylococcus. It is staphylococcus. Staphylococcus. What is this means? Staphylo means it is a bunch. It is a bunch like grapes. Bunch like grapes. So how the grapes are present? Now they are present like this. So in this way they are present, right? So in this way they are present. So in this way bunch of, what is this? Coccus is spherical shaped bacteria. Understood or not? So in this way if they are present, it is called staphylococcus. So in this way, based on the type of arrangement, so these names are given here, and this is based on the shape, this is based on the arrangement. Understood or not? Whereas some bacteria, so they are forming some three-dimensional structures. In that case we are using a word called as sarcina. Okay, so here they are arranging like this, so that, so in this way in the three-dimensional structures they are present. So in this way also different names are there, but anyhow these names are not that much important. My opinion or my interest is this particular one, because this anyhow we need to study under the experiment, and that is part of our NCERT. So there you need to understand this meaning. Coccus. What is the shape of Griffith's experimental organism? It is a question that is possible, and what you need to write, it is a spherical shaped bacteria. That's it. So for this one, anyhow the name is given here, and anyhow in this chapter we are talking about this particular shape of bacteria. Understood or not? So that is the reason why this will be in this way. If you are able to interlink the information, you can easily understand. So you don't need to by heart them every time. Simply understand the concept. Yes, example you need to remember, and remaining concept you can understand. So don't try to by heart the concept also. Okay. So this is what the information that they have given, and here this is the first paragraph, one paragraph that they have given on the prokaryotic cells. And after that in the next paragraph beginning what they are saying. So in the case of prokaryotes, different shapes are present. Yes or no? even though different shapes are present, basically almost the structure remains same. Okay, almost same means some definitely some small differences may be there. So you can say that similar with respect to the structure, you can say similar or you can say almost same. That means definitely some difference will be there. All the bacteria do not have the same properties, right? So they are having some small differences, that anyhow it is okay, but we are not worrying about those differences. Now at the level of class 11th and 12th, we are focusing on similarities, so that we will try to understand what is the average or the minimum basic structure that is present. So only that part we are studying. Anyhow, the differences, they are not needed at this particular level. In higher classes we will study. Understood? So in the higher classes you will study about differences. So now we need to focus on the basic structure. So that's why now we need to go to that, but before going to that, first we will complete the NCERT sentences for whatever we have studied till now. So in the case of prokaryotes, mainly we are talking about this bacteria we are talking, and cyanobacteria, that means blue green algae we are talking, and mycoplasma also we are talking. And regarding the mycoplasma, already I told you these are the smallest cells and they don't have cell wall, but anyhow we need to study about the cell wall. So when we are talking about cell wall, once again I will tell you about this particular point, and there we will cover. And within the mycoplasma there is another one that is called a PPLY is also there. That's what. And based on the shapes. So spherical shaped bacteria are called as cocci, and rod shaped bacteria are called as bacillus, and there are some vibrio is also there, and spirillum is there. So in this way different shapes are there, but apart from this, different others are also there. That's why we are saying that there are 4 basic types of bacteria with respect to the shapes. So these are the 4 different types that we are studying. Quickly we will go with NCERT sentences. The prokaryotic cells are represented by, means what are the examples for prokaryotes? Bacteria, blue green algae. Bacteria means eubacteria, archaebacteria, both of them will come under kingdom Monera only. They are considered as prokaryotes only. Next one, blue green algae, underline blue green algae and write cyanobacteria. Blue green algae are also called as cyanobacteria. And then mycoplasma and PPLO. PPLO are also part of mycoplasma. Please remember this, but they have given separately. PPLO, what is the full name? Pleuropneumonia like organisms. Clear? Next one. They are generally smaller and multiply more rapidly than eukaryotic cells. So they are comparing with eukaryotic cells. Smaller than eukaryotic cells. Already we have studied the size. So based on that we can say this. Multiply more rapidly than eukaryotic cells. So underline this particular multiply more rapidly, and just beside that in the free space you just write that. What you need to write? E. coli bacterium takes 20 minutes for division. Yeast, a fungus, yes or no? And it is taking, and that too it is eukaryote, it is taking 90 minutes for division. And then a human skin cell, that is also eukaryote, and it is taking 24 hours. Write the times there. So if it is less time, that means multiply more rapidly. If it is taking more time, multiply slowly. This is what the meaning is. So that's why you need to write that list just beside this. So in this way they are comparing with eukaryotic cells. Next one, they may vary greatly in shape and size. So they are having different shapes, different sizes are present. And the 4 basic shapes of bacteria. So though many shapes are there, 4 main ones that we are talking here are bacillus. What is the first one? Bacillus. Bacillus is what? Rod-like bacterium. And coccus is spherical shaped bacterium, and vibrio is comma shaped bacterium, whereas spirillum is spiral shaped bacterium. Understood or not? So these are the 4 different shapes they have given. Next paragraph. The organization of the prokaryotic cells is fundamentally similar. So if you are talking about the structure, whether it is rod shaped or spherical shaped or comma shaped or spiral shaped, whatever is the shape, basic structure if you are observing, it is similar. Even though small differences are there, majority of the structures are similar. Even though prokaryotes exhibit wide variety of shapes and functions, even though they are having different shapes and different functions, basically the structure remains the same. Understood? So now we need to talk about the structure. So in that structure also, first we will talk about the cell envelope. In that cell envelope, first we need to talk from top to the bottom, that means outermost to innermost. So in that sequence we are going. So before going with this particular information, so let us once again quickly revise whatever the overview that we studied in the previous lecture, so that we can continue this particular lecture. So first I will give the explanation. So once we are completing with the explanation, then I will come to this PowerPoint. Okay, now let us see here. So regarding the prokaryotic cell, in the previous lecture we studied about the overview. So in that overview, now here I will not write that entire one, simply I will draw the diagram. What is the overview that we have studied? So there is a plasma membrane here, yes or no? And outside the plasma there is a small gap, and here there is a layer, and this is called as cell wall. And attached to the cell wall there is a glycocalyx layer here, yes or no? In this way, what are the layers here? Outermost glycocalyx is followed by means next one will come. What is the next one? Followed by cell wall, and this cell wall is followed by another layer that is called as plasma membrane. It is called as plasma membrane, and this plasma membrane is also called as a cell membrane. Plasma membrane is also called as cell membrane. So these are the three layers, and these three layers combinedly it is called as cell envelope. It is called as a cell envelope. So in this lecture, first we will start with cell envelope. Anyhow, inside the cytoplasm, this is the cytoplasm. Inside the cytoplasm, fluid matrix is called cytosol, and then there is a DNA. It may be nucleoid or plasmid. Yes or no? Already we studied this. And apart from this, there are some organelles called ribosomes. And apart from this, there are some inclusion bodies. Yes or no? So in this way, we have studied about different things. And in this lecture, first we are talking about cell envelope. So cell envelope is having three layers. Outermost glycocalyx is followed by the use of followed by, follows, preceded by, precedes. Already we have studied in the general introductory lecture. So please refer that lecture first. Please refer that. Many students are making mistake in usage of these words. So please don't make such type of mistakes. Okay. So glycocalyx is followed by cell wall, and that is further followed by plasma membrane. So first we will talk about glycocalyx now. Glycocalyx, and it is present attached to the cell wall. Yes or no? And this glycocalyx, first we will see each and every word very clearly so that we can easily understand this particular information. This is a very small information regarding glycocalyx. What actually this glycocalyx layer is composed of? First we will see composition. What it is composed of? Understand the word here, glyco. What is this glyco? Many times we will study about this. Here we are talking about glycocalyx. And later on in the next coming lectures, we will study glycolipids, glycoproteins, proteoglycans. In this way, many times we will use this glyco. Yes or no? Glycosidic bond. Understood or not? So, glycosides. In this way, many names are there. So, whenever we are using the word glyco, it is a carbohydrate. It is a carbohydrate. Please understand this. Glyco word is used to represent carbohydrate, not just glucose. It may be any type of carbohydrate. So it is a glyco. That means this glycocalyx layer of the bacterium is made up of, or it is rich in carbohydrates. In the carbohydrates also different types are there. In the biomolecules chapter we will study about this. What are the different types of carbohydrates? Monosaccharides, oligosaccharides and polysaccharides. Within the oligosaccharides, 2 to 10 units are present. Monosaccharide means only single separate unit. Polysaccharide means more than 10 units are present. Yes or no? So in this way, many are present, and mainly these carbohydrates are different types of polysaccharides are present. It is having different types of polysaccharides. Name of these is not needed because in different bacteria, this composition is slightly different. But definitely carbohydrates are present. That is the reason why it is called as glycocalyx layer. Understood or not? So this glycocalyx is having carbohydrates. In that carbohydrates also mainly it is having different types of polysaccharides. Names are not needed. And not only this, some other small amounts of proteins and some other things are also there. Okay. So in this way they are present. So this is about the composition, because, and here they have mentioned it, layer name itself is glycocalyx. That means it is rich in. Whenever you are using the word called rich in means what? More number are, these are present. Not only this, something else are also present, but their content is very less. So those which are more, for that we are saying that rich in. Rich in polysaccharides means more number of polysaccharides are present, and the remaining content is very less. Some others may be present. This is what the meaning is. So glycocalyx layer is rich in polysaccharides, carbohydrates. That's why it is glyco and the calyx layer. Understood or not? That's what the name they have given. Fine. And this glycocalyx layer, it is having different thickness and it is having different composition. So that's why we are not going into those further details. And now here it is of two types. This point is important. It is of two types. This glycocalyx layer is two types. What are these two types? First one is slime layer. First one is slime layer and second one is capsule. 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 Please understand this. Slime layer is nothing but glycocalyx. Capsule is also nothing but glycocalyx. But when you will use the term slime layer and when you will use the term capsule, this is most important point. Understood or not? When we will use slime layer term, see, observe this. Suppose this is the plasma membrane. This is plasma membrane and around this cell wall is present. Cell wall is present. Fine. Outside the cell wall glycocalyx is there. And if this cell wall, if this glycocalyx is a loose sheath like structure, if it is a loose sheath like structure, sheath or sheet, anything you can say. So it is a loose and a sheath like structure. And if glycocalyx is present like this, then it is called as a slime layer. Now you can understand that a slimy, when you will say slimy, if it is sticky, yes or no? If it is semi-solid like and sticky, so then it is called as a slimy. Yes or no? So this layer is loose, a sheath-like structure. That's why it is called as a slime layer. Okay? And when you will say it as capsule, if it is thick and tough means what? It is very hard. If it is thick and tough, then it is called as capsule. So now this is the plasma membrane, this is the cell wall. Now around this cell wall, now attached to this cell wall, there is a thick and hard capsule is present. It is very thick and tough. It is very hard. Then it is called as capsule. And mainly, what is the role of this slime layer? What is the role of capsule? In the NCERT, simply they said that if glycocalyx is present as a loose sheath-like structure, it is called as a slime layer. Completed. Over. And here, if glycocalyx is thick and tough, then it is called as capsule. Over. NCERT sentence is completed. But we need to understand the function at least. What is the function at least? And here, since it is having this slime layer, now it will help in sometimes attachment. And many cases it will protect the bacterium. Glycocalyx, whether it is slime layer or capsule, in general function of slime layer is protection. It will protect the bacterium, not just the glycocalyx layer. Glycocalyx, cell wall, plasma membrane, all the three combined it is called cell envelope. So cell envelope general function is, or the default function is protection. They will give protection to the bacterium. Understood or not? And here specifically glycocalyx is resistant to different types of chemicals also. Okay, so this glycocalyx is resistant to chemicals also. So in this way, glycocalyx layer is helping the bacterium in protection from different chemicals also. Anyhow, normal function is general protection, and specifically it is giving protection from different types of hard chemicals also. Please understand this point. And this capsule, in very rare cases, this capsule is formed. And what is the function of this particular capsule? This capsule helps in protection of bacterium in adverse conditions. Adverse means what? Unfavorable conditions. When conditions are not favorable, there are more chances that bacteria may die. Understood or not? So in those cases, mainly in those cases, this bacterium is developing a thick and hard, tough capsule around this. Because of this, it can withstand, it can resist, it can even survive in extreme conditions, hard conditions, unfavorable conditions. And in this way also, this tough capsule is giving protection for the bacterium. This is what the overall function. So glycocalyx is having different thickness and a different composition, but mainly it is composed of carbohydrates, in that specifically polysaccharides. Clear? Next one. If glycocalyx is present as a loose sheath-like structure, it is called as slime layer, and a slime layer helps in attachment in some cases, and it is also protecting bacterium from some chemicals. Next one. Capsule. If glycocalyx is present as thick and a tough layer around the cell wall, then it is called as capsule, and the capsule generally protects the bacterium during unfavorable conditions. So once favorable conditions comes, this capsule will be removed. So in that case, now bacterium can once again start a division. So in this way, mainly the capsule is protecting the bacterium during unfavorable conditions. That's it. So this is the simple information that they have given in NCERT. With this, glycocalyx part is completed. and now we need to go for the cell wall, but regarding this we will see later on, and first quickly we will go through this particular one. If glycocalyx is present as loose sheath like, it is called as slime layer. If it is thick and tough, then it is called as capsule. That's it. Okay. Anyhow, regarding the cell wall and all, we will see in the next lecture. Okay. So this is where we are stopping. So in the next lecture, we will start with cell wall structure and what are the different sentences given in NCERT. For that understanding, please, I am telling you now itself, we need to go little out of NCERT, but ultimately to understand NCERT sentences. Okay. For the cell wall. Okay. We will meet in the next lecture. Thank you so much. Thank you.